So what I've done in part one was to try to describe for you, give you an overview of the anatomy, the physiology, and of one part of the visual system, namely the ventral stream of the visual cortex. What I want to do next is to show you how one specific can you hear me? Actually, I hear an echo. No. What I want to show you next is how a visual architecture, which is reminiscent of a lot of the deep learning architectures that you've heard about in this summer school, can be actually constrained, given available neuroscience data, to reproduce some of the data that I described. So one point that I want to emphasize from the start is that, as you can see, so this is a sketch of this architecture. As you can see, the arrows go from the bottom to the top. This is a purely feed-forward architecture. Again, as I alluded to in the first part, we're not going to try to account for natural everyday vision when you are free to move your eyes and shift your attention. Rather, we're going to try to account for these types of rapid categorization tasks and perhaps the first 100 or 150 millisecond of visual processing before cortical feedback become active. Okay. All right, so let me emphasize first that there's been a long tradition of researchers who have worked on this type of architecture way before I was even born. So this started with qualitative work by Ubel and Wiesel. I gave you a very quick highlight of their qualitative model in the first part. There were two notable computer vision systems that were developed. Fukushima was the first one really to take Ubel and Wiesel's ideas for building a hierarchy of visual processing and applied that to digit recognition. Jan LeCun was one of the first to get those types of architecture also to work. The architecture that I'm going to be describing is a close cousin of the so-called convolutional networks. All right, so I think the model that I'm going to be describing really is a system-level computational model. It's relatively large scale. It has on the order of 100 million units, which requires a little bit of hardware to simulate. Unlike computational models that tries to explain specific visual phenomena like contrast normalization or specific attentional visual search task, or try to model one specific visual area, the goal with this architecture is essentially to build what I would call a first-order computational model. This is a model that's going to make a lot of simplifications about actual underlying processings and computations, but the goal is to try to be able to simulate the system as a whole. Okay, so not just focusing on one area, but simulate an entire set of areas that we believe are critically involved in our ability to process shape information and recognize objects. So as I alluded to earlier, there are actually quite a bit of similarities, at least at a course level, between this architecture and kind of state-of-the-art computer vision systems. I mentioned convolutional networks, probably deep belief networks as well. And there's also related work on compositional hierarchies, work by Alan Ewell, Leonardis. There's work by, even at Brown, by Stu Geman and colleagues. So, this is really quite a generic family of models. One key difference between the work that I'm going to be describing and the work you might have heard about in machine learning and computer vision is the goals are quite different. So although the work that you've heard about, the goal is to build the best possible visual architecture, our goal here is to build the best model of visual processing. Okay, so we've constrained the architecture by the anatomy and the physiology, and most of our day-to-day job is to simulate electrophysiology experiments in this architecture and compare the behavior of units in different stages to those of cortical cells in corresponding brain areas. So the key idea is quite simple. The basic idea is a direct extension of Ubel and Wiesel, and the idea is that through specific selective pooling mechanisms done in the right way, we should be able to build gradually some increase in selectivity and invariances that has been observed throughout the ventral stream of the visual cortex. So the architecture starts with simple and complex cells, as described by Ubel and Wiesel. Just to refresh our memory, simple cells tend to be relatively small, fraction of a degree of visual angle up to a degree or two. They are sensitive to contrast, and in particular to the contrast between figure and ground. So some simple cells like a white bar on a black background. Some prefer a dark bar on a white background. So these are different populations of cells. They are orientation sensitive, and they are highly sensitive to the position of the stimulus within their receptive field. Okay? In contrast, complex cells tend to have receptive fields which are twice as large. Unlike the simple cells, they are invariant with respect to contrast reversal. So you can reverse the figure, ground, contrast, and the same complex cell will still respond in the same way. And most importantly, the complex cells tend to be tolerant with respect to the exact location of the preferred stimulus within their receptive fields. Okay? So from simple to complex cells, we've gained invariance or tolerance with respect to 2D transformations, namely position and scale. So the way we actually implement those stages is quite standard. A good model of simple cells is actually the so-called Gabor functions. You've seen a couple of examples of such cells next to experimental data yesterday in Bruno's talk. Here's just an example from early work by Jones and Palmer. Here they were fitting simple cells receptive fields to actual Gabor functions. Those are 2D Gabor functions. So these are the raw data. This is the fitted data, which is here is the residual. This is undiscriminable from noise. Of course, there is much more variety to that in the primary visual cortex. Again, we are hoping to capture some kind of first-order trends in the population statistics. So we have what I would call a battery of Gabor filters within each cortical columns. So we have a number of orientations, number of spatial frequencies, number of phases, which I'm not showing here. I'm only showing one phase, but in practice we have 4 different phases of Gabor functions. As I mentioned earlier, a hallmark of cortical organization is this idea of cortical columns, whereby the same basic dictionary of features, in the case of the primary visual cortex, we're talking about spatial frequencies and orientations, would be replicated at all position and scale. Okay, so we pretty much follow this architecture by replicating, this battery of Gabors at every position and scale. Okay, so from the computer vision perspective, you can think of that as a convolutional operation. From the neural perspective, we'd like to think of that as what is called a full replication scheme. So it's not like we are moving a kernel around, but we have the same basic neurons reproduced at every possible locations. Okay. All right. In terms of the C1 complex units, which is the next stage of processing, there are 3 key elements in being able to reproduce actual neural data. And we had this discussion during the break. I think it made me very happy yesterday when I heard Mark Aurelio mentioning that there were 3 stages, 3 kind of computational mechanisms that were critical to get good performance out of his hierarchical convolutional network. I think he mentioned pooling, squaring, and normalization. And as you'll see, those are the 3 key operations that I'll be describing here. Those are the 3 kinds of operations that have been described over and over again during the past 20 years that have shown to be critical in terms of explaining most of the tuning properties of cells, both in the primary visual cortex and beyond. Okay, so the first one, as I mentioned, is what I would call a half rectification, squaring operations. This corresponds to, at one specific location, looking at the output of a subset of the simple cells, all tuned to the same orientations, but different phases. Okay, and by taking a square of their responses, again, this is a model that has been shown to be accurate from the neurophysiology perspective, we are able to build complex cells which are invariant with respect to contrast reversal. Okay. The next stage that is critical is the so-called gain control divisive normalization. So I should, by the way, I forgot to mention that this was a model which is often referred to as the energy model that was introduced by Bergen and Adelson back in the 80s. The gain control circuit is a model that was described a little bit later by Eager and Carandini. The idea is as follows. One considers the response of a cell at a preferred orientation, 135 degrees orientation here. The basic idea is to normalize, that is to divide the response of this unit by the pooled sum response of cells at multiple orientation and phases. Okay. So whatever response this guy giving us, we're going to divide by the pooled activity. And so what Iger and Carandini have shown is that these types of neural circuits, which by the way would be implemented by a specific kind of division, which is so-called divisive normalization that can be implemented via shunting inhibition. And these pooling are critical if one wants to explain the specific tuning, the response properties of cells to change in contrast. The third mechanism that is critical are the pooling mechanisms that are giving us this increased tolerance with respect to 2D transformations. Okay, so in order to build some invariance or tolerance, I should say, to position, the basic idea is to wire together an array of simple cells covering a small part of the field of view. In this case, this would be, say, something like about twice the size of any one of this simple cell. They are all tuned to the same preferred stimulus. This is critical. And by wiring them together, it's possible to build a complex cell here, which will be tolerant with respect to the location of an horizontal bar anywhere within its receptor field. The reason for that is because all these cells have the same exact tuning properties. If I move this A letter around locations, it will activate a different afferent subunit, afferent simple cell every time. But because they have the same properties, we expect the response of this cell to be the same across locations. Okay. The effect is that this guy should have the same constant response irrespective of the location of the stimulus within the receptor field of the cell. And then similarly, the same ideas can be applied to scale. In practice, our complex cells pull over the activity of nearby spatial scales. And that accounts for the fact that there is a broadening in the spatial frequency tuning from simple to complex cells. Okay? And that can be very nicely explained by this local pooling over scales. Okay? All right, so the basic idea is to, extending these ideas from, simple and complex units from the primary visual cortex to extrastriate areas, so to area V2, V4, and PIT. By alternating between these two kinds of operations, what I'm going to show you is that it's possible to account for this gradual increase in the complexity of the preferred stimulus, as well as the gradual increase in the invariance properties that's known to be happening in the ventral stream of the visual cortex. Okay, so as I mentioned, we're considering two separate populations, functional types of cells. The simple units, we're assuming are responsible for increasing the complexity of the representation. The operation that they approximate is similar to a template matching. The way this is implemented is, and I'm going to skim through a lot of details here, but is via a high-dimensional Gaussian tuning. So by pulling together afferent units with different selectivity and the proper operation, it's possible to go from a simple representation, say to single orientations, to something more complex that's able to respond to complex combinations of those simple orientations. So from simple edge detectors, it's possible to build something like a T-junction detector, for instance. Okay? So you can think of this operation as a conjunctive AND-like operation, whereby those guys would be responsible for detecting conjunctions or the simultaneous activation by a bunch of afferent units. Okay. The complex cells are responsible for increasing the tolerance of the representation with respect to 2D transformations. We've made the assumption that the key operation is a max-like pooling operation. So I'll show you later some evidence for that, but I don't think it's critical to achieve actual invariance to position and scale. The basic pooling mechanism is to consider a population here of afferent S units. So this would be S units with the same exact selectivity. So that's, I think, the main difference between these two populations of cells. Those guys combine different afferent subunits. Those guys combine similar afferent subunits with the same preferred selectivity, but slightly different position and scale. One can think of this operation as a disjunctive operation and an or-like operation. Okay. So the point is to figure out that out of all the possibilities, at least one of those guys is activated in order to activate that guy here. Okay, so what are the evidence for these two kinds of operations? By the way, I'm I guess I'm making a strong point here in terms of assuming two functional types of units. In practice, things are probably much messier, and there's probably a continuum of response profiles between these two kinds of cells. But there is evidence for, yes?
I was just wondering how this is implemented in neurons. Is it just a different threshold for the cell, or how?
You mean switching between a tuning operation and a max? Right. So in the model implementation that I'm going to show you, I'm not going to care about biophysical implementations. I'm going to implement a max when I need a max and a Gaussian tuning when I need a Gaussian tuning. But there are biophysically realistic models to implement those. When you look at the actual circuits, they're actually exploiting divisive normalization. Well, essentially, I'll give you more details offline, but the flavor for this is to consider populations of cells on numerator and denominator with different exponent for the rectification function. So if you think of, for instance, a way to approximate a max or a softmax is by summing over, say, some x's to the power of 3, divided by the sum of the xi's to the power of 2, for instance. If one of the xi's is much larger than the other ones, you'll be able to approximate, say xj is larger than the others. This will be very close to approximating xj if xj is larger than all the other xi. Okay, so that would be one way to approximate the max. If you revert the exponent, for instance, if you rectify much more strongly, and this can be done by changing the synaptic properties of the properties of synapses. If the exponent is smaller than so the numerator is smaller than the denominator, then you can approximate something much closer to a tuning operation. In fact, people have shown that one can approximate a Gaussian function with such a divisive operation plus a sigmoid. Okay, so the short answer is that the same circuit can account for both. Those are just different parameters guiding the strengths of the rectification happening at the synaptic level. Okay. All right, so evidence for so-called tuning operations. So here's an example, standard example from the primary visual cortex. What you see here is the tuning curve. So here's the orientation of the stimulus being presented, response of a cell. You see beautiful bell-shaped-like tuning around preferred orientation at 0 degree here, and then you see that as the stimulus being presented rotates away from this preferred orientation, one gets a gradual bell-shaped-like decrease in the response. Okay. There are, beyond the primary visual cortex, there are many examples. I would say that this tuning operation is probably one of the most ubiquitous circuits in the cortex. There are evidence for this kind of tuning in higher visual areas, as well as somatosensory areas all the way to motor cortex. Okay? Here's an example, for instance, of tuning in the IT cortex. This is a relatively old studies from Logothetis and colleagues. What they did is that they trained a monkey to discriminate between various versions of these wireframe objects that they call paperclips. Okay, so the monkey was presented with two stimuli, one after another. First, a sample stimulus, which could be one of these objects. And then the second stimulus could be either one of the same paperclip but rotated away from the sample view, or a different paperclip. And the monkey was trained to report whether those two objects were the same or different. What you see is that after training the monkey, they started to record in the IT cortex and found a very large, significant fractions of the cells that became selective for these objects. I think they report more than 50% of the cells to become selective to these objects. And by the way, yet another evidence for learning and plasticity in IT, because it's very unlikely that the monkeys had any experience with these kinds of objects before they were trained. So what you see here are the tuning curves for 3 cells in IT. Blue, red, and green. What you see is that for every one of these neurons, there is a preferred view. So all of those are different paperclips. This is the preferred view of the paperclips. And you see that as you rotate away the paperclip being presented to this neuron, the response curve of the neuron kind of drops off with this kind of gentle bell-shaped-like tuning. Okay? So plenty of evidence for tuning. I think that's well accepted. Little less evidence is for this max-like operation, but there is evidence nonetheless. So here's an example of a study conducted in David Furster's lab a couple of years ago. The critical prediction for this max-like operation at the level of the complex cells is the predicted behavior when more than one stimulus is presented within the receptive field of the unit. Okay? The prediction is that if you look at the response of the unit, by the way, I should say that those are intracellular recordings, so you're not going to see spikes that have been filtered out. What you see instead is the voltage, the membrane potential. Again, the higher this potential, the stronger the response of the cell. What you see here is the response of the cell to any one of 5 possible stimuli. Each of these 5 stimuli correspond to a bar, horizontal orientation at 5 different locations in the receptive field. Okay, so you see that, actually the invariance of this, or the tolerance to position to these complex cells, is not perfect because, it responds much more strongly to a bar presented in this location than say at this location. Okay. Now, the critical prediction for the max operation is what happens when you present simultaneously 2 bars within the receptive field of the unit. For instance, if I present this weak, if I activate this relatively weak subunit versus a better subunit. Okay. Any kind of linear summation would predict a response which is larger than any one of the responses of the stimuli presented in isolation. An average would predict that the response of the cell falls somewhere in between the response of the 2 stimuli presented in isolation, and the max predicts that the neuron should only care about the best stimulus. And what you see here is an example, perhaps the best example of a unit computing the max. It's very hard to see, but trust me on that. They show that the response of the cell is best explained by a max-like operation. Okay? And it's not like they found this max-like operation in every possible complex cells that they recorded from, but they found that it was at least found in a subset of complex cells, which are the one that we need, by the way, for position and scale invariance. A little bit more evidence. Actually, I should speed up because, so I have until 12:30, right, Marco Aurelio? I have 12:30, right? Yeah. Okay, all right, so not that bad. All right, so further evidence for higher visual areas. This is an example from area V4. What they did here is a slightly strange experiment, I guess, which is they looked at the response of the cell when either one stimulus was presented in isolation or 2 stimuli were overlapped, so superimposed in space. And again, they found that a max-like operation is a pretty good approximation or predictor of the cell response when the 2 stimuli are presented simultaneously. All right, so as I emphasized already earlier, what we've tried to achieve with this visual architecture is to try to build what I would call a first-order model of visual processing. So we get rid of a lot of the details and try to constrain as much as possible the operations that I described, in particular the pooling ranges, say in lower stages, the frequency bandwidths, orientation bandwidths, peak spatial frequencies, et cetera, so that they would reflect what you would expect from cortical cells in similar areas. Okay? In particular, full disclosure, I actually didn't try to learn invariances in this architecture, and I hard-coded the invariances. I've been assuming that I've taken these cortical columns and the basic idea that the same dictionary of features would be replicated at all position and scale. I've hard-coded the network so that I would be able to build, design complex cells that would have just the right range of invariance properties, the right kinds of receptive field sizes, et cetera, et cetera. Okay? There is one thing we learn, and the learning procedure that we use for that is very, very simple. I'm almost ashamed to present that to a machine learning, deep learning network summer school. But this is the selectivity of the simple units. Okay? So the simple units, essentially, what needs to be learned is the preferred selectivity, the preferred features for those units. And the way we implemented learning is extremely simple. We considered random patches of natural images, and we presented those to the network, one layer at a time, intent style, and then we just stored those in the synaptic weights of the unit. So we built dictionary of units, of features, that were tuned to random patches of natural images. For those of you who are familiar with radial basis function networks, which are typically shallow architecture, the underlying motivation was kind of thinking about extending those to a hierarchy. Okay? So what's being done in radial basis function network is simply to put Gaussian bumps onto individual training examples, and from that comes the power by interpolating across those random examples. Now, we don't have enough space to store all possible random patches of natural images, so a shortcut we took was just to sample random patches, hoping we would still approximate the statistics of those patches. So it's still very crude. Now, in terms of explaining a high-level function, such as object recognition or rapid object recognition, we've been working under the assumption that what's happening when a participant is being presented with a stimulus is that it will activate a bottom-up, in a bottom-up fashion, a hierarchy of these visual features that I should say are shared across multiple object categories. And we've done experiments on computer vision databases suggesting that the architecture was able to generalize from datasets to datasets. So here's an example. For instance, I'm flashing an image here to this architecture. I'm detecting a loose collection of visual features across layers of this hierarchy. That in turn will create a population code at the top, say somewhere in IT, whereby each image will be associated with a pattern of neural activity. Okay. The assumption we are making in order to be able to categorize those images, and in particular to be able to discriminate between animal and non-animal, is that we have a little bit like the PFC unit that I showed you in the first part. We have a linear decoder available, something like a classification unit sitting somewhere else outside the ventral stream, maybe in the prefrontal cortex. There's evidence for category selectivity in the prefrontal cortex. Maybe in higher level memory-related areas like the hippocampus that Marco Aurelio mentioned yesterday. Okay, so that's the basic framework. Now, one criticism that comes back fairly often, at least from yes. Yeah.
Were you using random patches in all layers? Did you change the size of those random? How did you pick?
Yeah, so sorry, so I should clarify that. Yes, so the patches are actually scaled to the receptive field sizes of the unit. So, so essentially, we actually present a whole, very, very large image, and those images are shifted in, at multiple positions, and then the units during learning just gets imprinted. That is, the synaptic weights of the unit just store whatever activity, neural activity was generated at the stage just under by whatever image patch fell under its receptive field. So yes, as we go in higher and higher visual areas, the size of the patches, that are being used for the imprinting of these units will increase. All right, so one criticism that we get from, specifically from monkey electrophysiologists is the fact that we don't learn the invariances, and often people think this class of models as implausible because of the weight sharing and the assumption that you would have to replicate essentially or force the same weights to be shared across multiple locations. So we didn't implement the more realistic learning to be implemented at every stages, but we wanted to show at least that in principle it was possible to learn both selectivity and invariances of complex cells at the level of the primary visual cortex. So the basic assumption we have when working with these simple and complex units is that the simple units would be essentially detecting correlation in space. That is, they would be detecting the kind of stuff that are happening at the same time over different locations. Okay, so for instance, one way to learn a T-junction is by learning that a T-junction is made by the simultaneous activation of afferent units tuned to a vertical, and a series of horizontal bars. And so in practice, this can be very well implemented via simple ABN learning-like mechanisms, whereby the kind of stuff that gets co-activated between afferent units and postsynaptic units gets the synaptic weights of the connections get increased and decreased otherwise. For complex units, we have actually been building on a long history of modeling work, starting by the work with Peter Foldiak in the 90s, who have suggested that one way by which these invariances could be learned is via transformation sequences. Okay, and so the learning rule we have implemented was a version of a ABN learning rule, whereby instead of reinforcing synapses between presynaptic sites and postsynaptic sites of things that are co-occurring at the same time, we introduced a delay between the two. What that delay allows us to do is when a pattern is, for instance, translating in front of the receptive field of a complex cell, it's going to activate a sequence of simple cells. And with the proper time constants for the synaptic weights between the simple and complex units, it's actually possible to learn to wire together patterns that are happening in nearby temporal continuity, which, in general in the natural world turns out to be the same kind of feature that has been translated or slightly changed in terms of its scale. Okay. So this is just to, very quick, to show you the kinds of things we can learn from using these very simple learning rules, starting with a population of on and off center-surround receptive fields at the level of the LGN. We essentially used a natural movie. This is, I don't know if anyone has seen this movie. This is actually a cat cam that was collected in the lab of Peter Koenig in Switzerland. This is a camera that's been put on the head of a cat, and the cat is just let run loose in the woods. And I guess this is the Swiss version of natural movies. Of course, there's a lot of jitter because there's no camera stabilization. Anyway, we needed a natural video sequence. We used it. What I'm showing partially here is that we're able, to learn simultaneously the selectivity of simple cells. These are the kinds of orientation selectivity we are getting for every one of these locations. And I should emphasize that those are learned independently for every one locations. And we further showed, this is not, I'm not going to have time to show you the results, but we showed that using this variant of an Abian learning rule, introducing delays between the presynaptic and postsynaptic sites, we also showed that it was possible to learn the right kind of wiring from simple to complex. And the right kind of wiring is learning that this guy, which should be tuned to a vertical edge, should pull together, from within every subset, every cortical column, only the vertical bars that are being presented at that locations. And we verified, and the tuning properties of the complex cells we obtain matches those of cortical complex cells. All right. So part of our everyday job, as I mentioned earlier, is to test these models against Christian. Yes.
So you just mentioned the time aspect before, and how did that come in the video and the cells you got in the tuning?
So I see. So time in the video. So in particular, the speed. Sorry, the speed at which things are changing will have an impact on transmission at the level of synapses here, right? So essentially, this guy is going to constrain what we can consider being continuous based on the synaptic conductances here and the delays introduced. So I'm not sure. It doesn't look like I'm answering your questions. So essentially, we're presenting this network with frames one after another. And then for every frame, we're updating the synaptic weights of the synapses. And again, the critical kind of parameter to be tweaked is that the synaptic delays here should be matched to statistics of natural images. You won't be able to learn slow translation, for instance, with very fast synaptic weights here. So somehow the two have to be matched. Okay. I'm happy to give you more details here if you want offline. Again, we haven't implemented that throughout the entire visual hierarchy, but I think in principle there is nothing that would prevent us from actually applying this learning rule for simple and complex cells throughout the visual cortex, or throughout the model of the visual cortex. So as I alluded to, we spent a fair amount of time comparing the tuning properties and conducting simulated monkey electrophysiology experiments on model units, right? So we play the game of considering random populations of units in different stages of this model. And then we present the same stimuli in conditions which are as closely related, as similar as possible to what is done in experimental conditions, and compare the tuning properties of the two. So we spent a fair amount of time doing that. I just want to give you a flavor for the kind of tuning you might get from intermediate stages of this hierarchy. What I can tell you is that despite the simplicity of this visual architecture, despite the fact that we are neglecting on a lot of complexity and known nonlinearity at the level of dendrites and everything, so far we found that this is a good first order model. And what I mean by that is that if you take a random population of cells in one stage of this model and compare it to a random population of cortical cells in the corresponding brain area, we find that in general the distribution of cells or their properties are indiscriminable. Now, I'm not claiming that this is the only model that could account for that. In fact, and we can talk more about it offline, I think those data are not constraining enough for computational models. So I'm sure there is a whole zoo of models that could explain those data. But I think one can think of this model as a good way to to capture kind of the essence of a visual representation in terms of its complexity and range of position and scale invariance throughout stages of processing in the visual hierarchy. Okay, so here's one example out of several of a comparisons or a test we've done on this model. So there are data from Pasupati and Connor dating from a couple of years back. They were interested in looking at the tuning for boundary conformations and curvature computation in intermediate stages of the visual cortex. Okay, so the underlying model was an explicit model of explicit boundary and curvature computation. So they use this very specific set of parameterized shapes. So it would take me a little bit of too much time to explain you the details of how this is parameterized, but just to get a feel for it, you see that going from left to right, you're gonna be having an impact on essentially the level of curvature and the location of concave and convex parts on this visual stimuli. So their hypothesis was that explicit curvature computation is happening at the level of these visual areas. We have a very simple model, a model which is probably wrong, but a model that knows nothing about boundary conformation and curvature. We have a model that predicts that at the level of intermediate visual areas, such as V4, we should get a subunit organization that looks something like that. So what I'm showing here on this 3x3 grid is essentially the subunit organization of nine distinct cells in intermediate in area V4. Okay, so what I'm trying to show with ellipses is the preferred orientation of that specific subunit. And as you see, sometimes the subunit might have multiple preferred orientation at that location. I should also emphasize that this depiction is misleading because remember that the afferent subunits of each one of these subunits are complex cells which already exhibit some tolerance with respect to position and scale. Okay, so although I'm showing an ellipse at the center of the subunit, in practice there is a whole null space, a whole set of stimuli for which the cell would respond in much the same way. Okay, so there is almost an infinite number of stimuli that could activate this unit in an optimal way. Okay, and the colors here indicate the strengths of the connection from the afferent complex cell subunits onto this V4-like S2 units. Okay. So you'll notice that here I'm very conscious about not saying excitation and inhibition. This is due to the fact that we have this divisive normalization. And instead, I'd rather speak of facilitation and suppression. What that means is that when something is red, that means that putting this specific orientation at that specific location or within this subunit will tend to increase the response of the S2 units, while something that's more blue will tend to reduce the response of the units if this subunit is being activated. Okay. So again, we've done more quantitative comparisons. I can point to you the papers if you're interested in. Just want to give you a sense of the kind of tuning behavior we can get from this model unit. So here on the left was Pasupati and Connor's best neuron. Sorry. So this is a neuron that's strongly tuned for curvature here on the right side. By the way, I should say that the stimuli are faint to see, but the color on the background for each stimulus represent the strengths of the neural response for that stimulus. So dark means that the response fires much more vividly for that stimulus than for a stimulus for which the background is white. Okay, so this guy essentially likes curvature here on the right side. You see that this is shared with this stimulus here. It doesn't care so much about what's going on the left side there. Again, it's very hard to figure out what's going on. One interpretation would be that it computes curvature explicitly, but if we just take a very small population, I think in that case we took 100 model units at random, and just out of this population of model units, pick the one that best correlated with the response of this V4 neuron on that set of stimuli, and we came up with this unit, okay, which is a unit that has the following subunit organization, combination of two orientation here, horizontal here, diagonal here and horizontal here. And just to give you an idea, the correlation between these two was highly significant, somewhere on the order of correlation of 0.78, which, believe me, for any kind of neuroscience data, this is quite a large agreement. And so we've done more quantitative analysis. We've shown that at the population level, the model units that we produce with these very simple learning rules and computations is indiscriminable from the data that were collected for this experiment. Okay. Quickly, another experiment, very quick. I mentioned this experiment earlier. I just want to show you that this was actually a prediction from the model. So I mentioned this monkey electrophysiology recording done at MIT in Jim DiCarlo's lab. They showed that it was possible to decode invariant object category information from a small population of about 200 model units. At the time, Gabriel was involved in the analysis of data, was in the Poggio lab, sitting next to me. He actually tried to run the model before they actually started to record from monkeys. They had selected the set of experiments. He checked that the model was able to do the task from a small population of about 200 model units. And because the model was able to do that relatively well, they went on and did the actual recording. And you see that the model is a little bit optimistic about the level of performance, but the range of invariance to position and scale is actually strikingly similar. Again, there is no tuning here. There is no free parameter. It's not like we tweak the model to account for those data. These come for free from this basic architecture. Okay, so last point I want to make is that, of course, it's possible to simulate the overall architecture, the 10 million units, and it's possible to train a linear decoder on the population responses in these model units and try to predict the presence or absence of animal in visual presentations. We actually did the comparison to human observers. This is work that I did a couple of years ago as part of my PhD thesis with Aude Oliva at MIT. And so we ran human observers on a version of the rapid categorization task that I showed you before the break. To make the task a little bit more interesting and non-trivial, we actually subdivided our set into 4 subsets. Each subset corresponding to a different distance of the animal from the camera. So our first category that we dub head correspond to a big animal head. We expected that it would be relatively easy because salient features, pretty big animal. And then we considered animals a little bit further away from the camera with this body category. Should still be easy because most of the image is taken up by the animal, but one can start seeing a little bit of clutter. Then what we call the medium far category, whereby the animal is further away from the camera, and actually maybe in the back you're starting to have trouble to see the animal here. And then the far body and groups, actually most of the image is taken up by clutter. The animal end up being a small portion of the visual stimulus. I should say that we actually used a separate set of subjects to estimate the distance of the animal from the camera. So this is a completely objective assessment. And to make the task more interesting, we used a matching set of distractors. So for the distractor non-animal images, we used objects that were at a similar viewing distance as compared to the animals. Okay, so our hope is that there is no kind of simple low-level cues that will allow you to do the task. That you need to rely on a high-level, visual representation. So what is shown here on the x-axis will be performance broken down according to the 4 different subcategories. On the y-axis is performance as measured by the D prime. The D prime is a standard psychophysics measure which combines a heat rate and false alarm rate into one single score. The higher the D prime, the better the performance. So what you see here in blue correspond to the performance of human observers on each one of these 4 subcategories. In red is the model performance. As you can see, first thing to notice is that both of them agree relatively well. I guess it's probably luck on our side. But I think the most interesting result that came out from this study really is this, the fact that both human observers and the model do quite well on the 2 conditions that correspond to animals being very close when the amount of clutter is minimal, and this is the case for this head and body. But then as the amount of clutter in those images increases from body to medium far and to far and groups, the decrease in performance is very drastic. So just to maybe error rate will speak better to a computer vision audience or machine learning audience that D prime. Here the level of performance is about 90% accuracy. At the level of far, it's down to about 70%. So this is a drop of about 20% in accuracy for both human observers and the model. Okay. So this, I would argue, is a serious limitations of these types of feed-forward processing and hierarchical architectures. And I'll get back to that tomorrow when we talk about attentional mechanisms and top-down signals. Okay. All right, I want to spend the last 10 minutes or so giving you an overview of some of the work that's ongoing in my lab as we speak. And so the basic idea is, I would say, kind of a grandiose scheme. The basic idea would be to build a complete model of visual perception. And of course, that will never happen, but it's a worthwhile goal by itself. And so our work for the past year or so has been to, starting from this basic visual architecture, to try to extend it to include processing along multiple dimensions. And I'll try to highlight a couple of those extensions in the next couple of slides. The basic, kind of the basic ideas behind these extensions is something that was actually suggested and proposed many years ago by Adelson and Bergen. And this is the idea that the primary visual cortex and these kinds of oriented structures, Gabor-like structures, doesn't have to be limited to processing of spatial orientation. So for instance, one can imagine processing along multiple dimensions. You could imagine building Gabor filters that are not just tuned to x and y, but they could be tuned to, say, x and time, right? So they would measure the amount of change in the x direction across point in time. So that would be one way to detect motion, for instance. Similarly, one could detect changes along, say, u and space, or across depths and space, and so on and so forth. Okay, so the basic idea is to extend this original framework to take into account multiple dimensions. And so some of the work that we started with was to extend this architecture to include color processing. And the basic idea is quite simple. We know that in addition to the luminance-defined Gabor functions, which are the ones we used in the architecture that I just described, there are also oriented cells in the visual cortex that are sensitive to color opponent channels. Okay, so rather than having on and off subunits here that are based on the amount of light that just the luminance that's imprinting those subunits, people have shown that there are oriented cells for red and green opponent channels, as well as yellow and blue opponent channels. So those are relatively well established. We've also included a red sign channel, which is a little bit more debated. This was suggested by Bevel Conway, who used to be at Harvard and is now at Wellesley College. There's a little bit of debate about this red sign channel because it's not clear whether it's an actual channel on its own or a dirty, unquote, red-green channel where the green surround here has been contaminated by some blue. Anyway, in practice, it doesn't make a big difference. The point that I want to make here is that it's possible to analyze color images in much the same way, using essentially recycling the three key computational mechanisms that I described earlier, namely putting over space, half squaring over multiple phases, and divisive normalization to account for contrast responses. Okay, so we essentially implemented this hierarchical color processing with this hierarchy of processing stages. We actually tuned the parameters. There are a couple of parameters regulating, for instance, the strengths of the half squaring here, or the strengths of the divisive normalization, or considering different pools of cells to use to normalize. So we optimized those parameters in order to be able to reproduce psychophysics data. And in fact, we showed that with only two stages, hierarchical stages of processing, we were able to explain almost perfectly human data related to similarity ratings across views. Okay, so we had access to data from human subjects as to rate the similarity and dissimilarity across patches of views. And we showed that this simple extension was able to account for the actual similarity judgments from human subjects. Okay, so we were able to build a model of color processing that mimics very closely what the human visual system would be implementing. And of course, once you have a model of color processing, you can use that as a front end to any kind of off-the-shelf computer vision system you want. So we've extended our HMax model, the model of visual processing, to not just include Gabor filters tuned to luminance, but also to include these oriented color channels. In addition, we also, just to show the generalizability of the approach, we also use that in conjunction with other methods, such as the sift. And as much as we can apply these color opponent channels and rectification and normalization on the Gabor filters we use in the HMAX, you can apply just the same thing on the gradient-based filters that are used in the sift. You can also use that on Gaussian derivatives, which are used in the Berkeley system for segmentation, which is state-of-the-art. Okay? And again, we've used just the same exact starting from these filters. We use this hierarchy of processing stages. Here, what we are showing on a set of standard color data sets, which is the soccer team and flower, we are showing essentially the benefit of this, essentially respecting the processing in the visual cortex as a preprocessing stage. And so if you compare what you get from a sift descriptor that's using this color representation as input, as opposed to not just grayscale sift, but previous color extensions of SIFT that had been proposed. So previous extensions had relied on the gradient computed directly on the image of U and on a retinal, very simple opponent channel with no nonlinearity. And you can see that using this biologically inspired computational mechanisms, the level of performance of the SIFT descriptors on these color data sets is much, much, much higher than previously proposed descriptors. Interestingly for us, something we knew, but we still have trouble convincing people of that, we found that the HMAX-based representation actually significantly outperformed the SIFT descriptors on this task. We've also evaluated the performance of a simple kind of spatial pyramid. This is implementing the Lazebnik's spatial pyramid on the Pascal challenge. We're not, of course, claiming state-of-the-art because typically the state-of-the-art relies on a number of various descriptors and the combination of multiple kernels and heavy machine learning. But here, what we're showing is that we can report very significant improvements, almost 7% improvement from using this single opponent, double opponent SIFT as compared to a grayscale SIFT. In all fairness, it turns out that the HMAX on this task is very similar to what you would get from a bag-of-word model on the Pascal challenge. Okay, more qualitative results. I mean, again, we have more quantitative results. I should say that this is a paper that's going to appear in the upcoming ECCV. I believe that this kind of color representation can be quite useful for segmentation tasks. So here, this is showing essentially the hard assignment for every patch of the image here as measured by, so essentially mapping every patch of an image to the nearest entry in the code book of textons that was learned by the Berkeley system. And I should say this is the state-of-the-art system on this database. Okay, so the color don't mean much. These are pseudo colors. But what you should see here is that, for instance, the fur or the flock here of the school of fish, sorry, you see that there is a lot of noise essentially. So the grayscale texton-based representation is a poor representation, a perceptual representation that essentially treats all these pixels as very dissimilar. In comparison, if we extend the texton representation to include our color front end, you see that perceptually things tend to group much better, with nearby pixels tending to have the same hard assignment on the codebook entries here. So far so good. All right. One very recent extension, and this is still very much work in progress, is to include this model to include binocular disparity. Okay, so everything that I've shown you is based on the assumption that we have monocular presentation of stimuli to only one eye. So, actually relying on very similar computational mechanisms to the one that I described, things like squaring and normalization, it has been suggested that it was possible to essentially build complex cells that would be tuned to various disparity by combining the signal from one Gabor filter on one eye and the signal from another Gabor filter on the other eye. Okay? The trick is, in order to explicitly compute disparity, is to rely on either shift in position between these two Gabors or a difference in phase. Okay. So this is a model that's relatively well established and has been compared to electrophysiology data. This was proposed by Kian a number of years ago. we essentially extended this model and used that in conjunction with the simple and complex units in our model. Okay, so now in terms of learning of this architecture, rather than monocular patches of images, we presented the model with simple binocular patches of images. This is just obtained from a simple consumer-grade stereo camera. Okay, I had an honor student working on this. This was in his honor thesis, Guillaume Reason. Again, these are still very preliminary, but what he showed is that at least qualitatively, we're able to reproduce data that were described by Sasaki et al. From area V4. So what we are showing here is that we're able to obtain some orientation for our V4 binocular cells, tuning for 3D orientations. So what Sasaki and colleagues did in the experiment and what we reproduce to the dot in our simulation, it was first to map out the preferred 2D orientations of the cells. So first you find cells that are tuned, you find the preferred 2D orientations, and then once you find the preferred 2D orientations, you start changing the 3D orientations. And what you see here are tuning curves for orientation in 3D. Okay? And you can see that a lot of, not all of them, but those units that were trained from patches of natural images, actually a significant number of them exhibit this very nice Gaussian tuning for in 3D. Okay? And so what I think this suggests is that, little by little, we'll be able to extend this architecture to a much richer visual description than what's available from simply 2D-based texture information. Another extension very much in progress has to do with motion processing. So maybe I'm going to skip that in the interest of time, but coexisting with cells that are tuned to spatial orientations in the primary visual cortex are cells tuned to various directions of motion. Okay, so rather than being cells that are tuned in just space and space, they are tuned in space and time. So you can think of those as 3D filters, essentially, or 3D convolution in space and time. The way to implement those in biology is fairly well established. One needs non-separable space-time receptive fields. There are several models that have been proposed to account for that. What we've done is, and what actually is interesting is that although these two populations of cells coexist in the primary visual cortex, it turns out that they project to two different streams of visual processing. So the cells tuned to orientation in space project along the ventral stream to higher areas of the ventral stream, whereas those tuned to motion directions project to the dorsal stream of the visual cortex. So what we did here is essentially, again, apply some of the very same computational mechanisms, starting from the space-time receptive fields as opposed to simply oriented receptive fields, and build an architecture that we actually match to the organization of the dorsal stream. So I should say that the specific parameters such as the range of pulling ranges, the range of invariances, receptive field sizes, etc., differ a little bit between the ventral and the dorsal stream. For instance, in the dorsal stream, receptive fields tend to be larger, but we showed that it was possible to actually tune this architecture and explain for a host of data. And we're very much in the process of writing those up. Paper we published a couple of years ago testing this model of the dorsal stream for the recognition of human actions. Back then, the system was state-of-the-art. By now, there are many systems that are outperforming it, but still a couple of years ago, it was still among the top contenders. We are so happy about the performance of this model of the dorsal stream for the recognition of action that we decided to actually use that in a real-world scenario for the automatic monitoring and analysis of rodent behaviors. The reason we care about that is because a lot of my colleagues in the neuroscience department care about understanding the link between genes and behavior, and so that takes a lot of work from a lot of undergrad students to painstakingly annotating by hand every frames of videos over extended periods of time. There are many issues associated with human manual annotations. We've heard about scandals recently at Harvard because these things can get quite subjective, and also that limits our ability to conduct experiments over extended periods of time, because typically what human can do, it takes about 20 times more longer to annotate a video than it takes to actually record, right? So 1 hour video takes 20 hours of manual work. And so that limits the temporal extent over which we can conduct those tests. So what we did is A couple of improvements. We've first ported all our architectures to GPUs. That allows us essentially, with the current computational power of GPUs, to run on a single machine, analyze the behavior of 4 cages at the same time, where every cage, so this is one camera, 4 cages, each cage is being sent to a different GPU, and we're able to achieve real time on this setup. Again, very briefly, this is the orientation or the response of our population of V1 motion sensitive cells, color-coded, so colors match to different directions of motion. The reason I'm showing you the output of our primary visual cortex model is because showing you what's going on in higher stages is actually quite tricky. Anyway. I'm not going to show you the video to save time. What I wanted to tell you is that we're able to conduct this automated analysis in real time and at a level which is indiscriminable from human manual annotations. And this is true for about 10 behaviors of interest, things like eating, grooming, walking, hanging, jumping, and things like that. By the way, as a side note, I think this is actually an interesting problem for computer vision because it has some of the flavor of what we would expect from an actual, real-world computer vision problem. We are expected to generalize over multiple animals. Not all behaviors are identical, so we need to generalize over different types of instances of behaviors. But what I think is interesting is that it saves us a lot of the kind of pre-processing that as high-level vision scientists we really don't want to deal with in general. So in other words, we have good control. The camera is perfectly static. The illumination is very well controlled, et cetera, et cetera. So in my view, that allows us to tap directly into some of the high-level visual recognition problem without having to deal with nitty-gritty kind of low-level image processing. By the way, I should say that we're in the process of developing a high-throughput core where we have 48 cages that are being monitored 24/7. We are generating an insane amount of both unlabeled and labeled data, and we're making those data available. So if anyone is interested in testing various visual architecture, there is a lot of room for improvements. In particular, I'm quite convinced that given the fact that the problem is relatively well constrained, we should be able to do much better than human observers because this is a task that is very tiring, and most of the errors from human annotators are actually due to distraction and the fact that they get tired, turn their head, the mouse are really quick, they jump, and that's it, they missed it. All right, I think I should wrap up. I just wanted to point out that we have also used this model of the dorsal stream as input to a model for the control of target reaching and of avoidance of obstacles. I'm not going to have time, unfortunately, to give you too many details about that. But the take-home message that I wanted to give you is that a model purely based on motion processing in the dorsal stream, used as input to a model for the control of locomotion, is able to reproduce almost perfectly human data collected in a virtual environment lab. And I should say that this is a collaboration we have with Bill Warren here at Brown, who is working with virtual environments. And the work that we're doing right now is trying to essentially scale up this architecture to more and more complex visual environments, which, by the way, I think would offer a somehow interesting and natural way to train some of this deep learning architecture, where we would leave the autonomous vision system simply navigating in those environments, as opposed to simply showing them, YouTube movies, for instance. All right, so I'm done. What I've tried to do in this hour or so was to convince you of the fact that feed-forward hierarchical learning architectures are consistent or seem to be consistent with what is known about the anatomy and the physiology of the visual cortex, at least at a course level. And they also seem consistent or sufficient to explain human psychophysics during rapid categorization tasks. Of course, those are incomplete model of vision, not only because they are neglecting a lot of important details, but most importantly because they do not take into account the cortical feedback, which we know are massive in our visual cortex. Particularly, I've highlighted the fact that they are quite limited in terms of what they can, or at least what this model is able to do when dealing with natural images. It suffers from this clutter problem, which is something that people know already in computer vision, which is a problem with bag of words approaches. But I would say, more importantly, they're highly limited in terms of the representational power that they have for natural images. So in particular, they wouldn't be able to parse and interpret complex visual scenes. The point that I'd like to make, and I'll give you more details about that tomorrow, is that cortical feedback and attentional mechanisms would be needed to extend this type of architecture to allow them to start performing more interesting visual tasks. So, probably, I don't know how many of you are in fields related to biology. Probably many of you don't really care about biology. So if I have to give you one take-home message, it's perhaps to try to convince you to keep an eye on biology. And part of the reason for that is that, I think computer vision really used to be the driving force in terms of making predictions for neuroscience. But I think in recent years, neuroscience has been a lot of progress. We are developing almost everyday new techniques that are allowing us to understand in much greater details the behavior of individual neural circuits. So I believe that in the course of the next couple of years, biology is going to start to make more and more predictions and useful suggestions for computer vision. In addition, irrespective, as we've seen in the past couple of days, of course, in general, it's possible to learn everything end to end, right, from front to back. And that's something that many people here in this room are familiar with. But as we've seen, the space of parameters is actually quite huge. And if anything else, the argument I'd like to make is that it's possible to use, the state of neuroscience is good enough that it's possible to use data derived from neuroscience as at least an initial starting point for constraining the parameters in those algorithms. And I'm not excluding the fact that you could then later refine with machine learning techniques to find something that more specifically suited to your problem, but I think it could be at least a good starting point, which is what I try to do today. All right, so that's it for today, and we'll talk about cortical feedback tomorrow. Thank you.